Our last presenter, last but certainly not least, is Dr. Vidar Garg, coming to us from Kat Hajantanakis' lab. And his presentation is on how stem cells get their groove, looking for potential in all the right places.
So I want you to try to remember yourself in your first week of college. Maybe you can hear some Green Day playing in the background. But you're coming in with a lot of optimism and potential. Now, if you were like me, you knew you wanted to be either a scientist, a doctor, or maybe an architect. My sister, on the other hand, she wanted to go into marketing. So how did we come to have such diverging interests? Well, for that, you'd have to look at our experiences before we got to college. So I had a really great biology teacher in high school, while my sister had all the people skills. So college perhaps gave us the means to pursue our interests, but it was really our prior life experiences which gave us that potential that we walked in with. Now, this is similar to embryonic stem cells. These are cells that are derived from the embryo, and they are pluripotent, which means that they can become any cell type in the body, whether that's a brain cell, a heart cell, or a liver cell. So this has immense therapeutic potential because, in theory, we can take these cells, we can grow them up in a dish, and we can make them into any cell type that we want. But in practice, this gets tricky because not all cell types are easy to make. And we're still trying to really understand what it means to be a stem cell. And so to bridge this gap between theory and practice and really tap into their potential, I'm looking back at the embryo where these stem cells originally came from. And I'm trying to understand how they came to have that potential that they do. Because their sister cell doesn't have the same potential, even though they grew up together. And so I can ask, what would it take for me to make the red stem cell into a blue sister cell, or take the blue cell and make it red again? This is similar to asking what characteristics would I need to have, or what would I need to change to be more like my sister, or vice versa. I can turn on a specific gene in the red cell and make it blue, or turn on specific genes in the blue cell and make it red again. And when I did this, I actually found that it's very easy for the red cell to become blue, but extremely difficult for the blue cell to become red again. And that's because stem cells are very flexible, and they can turn into anything that they want. But the blue cell has already made up its mind, and it refuses to change. And so I'm using single-cell genomics to try to understand what it is that makes stem cells so flexible. I found that on a molecular level, stem cells are more like wet clay. They can be molded into various shapes. But the blue cell is more like hardened clay. I've identified a core set of genes that really keeps the blue cell locked in its fate, and these genes are inactive in stem cells. Now I'm on the hunt for additional factors that keep stem cells so flexible, because now that we know where to look for their potential, we're really beginning to understand how it is that stem cells get their groove. Thank you.